So let's classify bacteria. We have discussed gram negative bacteria. Now discuss the gram positive bacteria. So gram positive. The first line of bacteria identification is gram staining, and if the gram dye stained when the bacteria or the respective specimen has a thick peptidoglycan, and it can give a purple or blue indication or colonies. So first one, there are three kind of bacteria. There is the bacilli, there are the filaments, and the cocci. So first one we discuss the cocci. There is a cocci bacteria. So the cocci bacteria, we have the first test to differentiate different types of cocci. We give catalase positivity. The structures, the bacteria, the cocci bacteria, the gram positive cocci bacteria, which give catalase positive reaction, are the staph family, which give clusters. So the staph family which gives clusters, and the staph in the next test is coagulase, and the structure which give coagulase positive is aureus, golden pigment, beta hemolytic. So staph aureus is coagulase positive. The coagulase staph negative organisms will do another antibacterial sensitivity test, that is coagulase negative novobiocin, and there are two conditions. Either the respective organism is resistant, or either it is sensitive. So the sensitive one is epidermatitis, and the resistant one is saprophyticus. So sapro is novo. So we differentiate three kinds of staph bacteria. First one, catalase positive staph. The staph, then we do a coagulase test, and coagulase positive is staph aureus, and then we give a novobiocin antibacterial test. The sensitive one is epidermatitis, and the resistant one is saprophyticus. Then we have the cat negative. Cat negative are the strips or the chains or the pairs. So they give strip, chains, and pairs. In the streptococcus bacteria, streptococcus family, we then give blood agar hemolysis. We do blood agar hemolysis, and in this hemolysis we have three kind of results. Either there is hemolysis or not, and if the hemolysis, is it complete or incomplete. So first one is alpha, which give partial hemolysis, and the color green color. Then we differentiate the alpha hemolytic catalase negative streptococci into then we give another test for optochin sensitivity or resistant, or bile soluble or not. So those bacteria which are, so there is a mnemonic over here, overpass. This is the optochin. Viridin is resistant, and strep pneumo is sensitive. So the sensitive one are the positive, or sensitive one are the strep pneumo, and they are also bile soluble, and the resistant one are the viridins, or they are not bile soluble viridins. Also there is strep mutans and step militans. So these are the alpha hemolytic bacteria. Then we have beta hemolysis. Beta means complete hemolysis, and they give black clear C4 complete, C4 clear or pale colonies. Then we do another test for bacteriocin, bacitracin or PYR stat. So those bacteria, there is a mnemonic here, B-Bras bacteriocin test, and group B is resistant and A sensitive. So the sensitive with the positive PYR test is group A streptococcus, for example group A pyogenes, and then the resistant one are the group B strep, they are A galactia with the negative PYR. Then there is this is all about hemolysis, and there is also another test for hemolysis. If there is a no hemolysis, then it is called gamma hemolysis, means no hemolysis. And if no hemolysis, then we do growth in 6.5% of NaCl or in bile, and we see their solubility or growth. So bacteria which give this kind of solubilities are so bile is present in our intestine. So those bacteria which live in our intestine are enterobacteria, means they are enterococci. So the family which gives this growth or a positive test are enterococci, and in this enterococci family we have the enterofaecalis, faecium and enterofaecalis. So this is a pretty much word to word literal meaning of this family. And those which are not, they are non-enterococci, not present in the not enterococci. So in this example we have the strep bovis, which can cause infective endocarditis or heart problems. So these are all about this is all about cocci, gram positive cocci. The next one is cocci. So the next one is, there is bacilli. So in the bacilli we have some classification for bacilli. There are aerobic, or they are anaerobic, and also there is a filamentous bacteria. So aerobic. In aerobic we have listeria and bacillus anthracis and bacillus cereus, and also there is Corynebacteria bacteria. And in anaerobics we have the Clostridiae or the Propionibacterium or the Actinibacterium. Then we have filamentous branching gram positive bacteria, in which there is also we do aerobic test, aerobic or anaerobic. So these two. So in the aerobic one we have the nocardia, and also we have the actinomyces israeli. We also do acid fast, so nocardia is weakly acid fast. So this is the general classification of bacteria. There is also some other small mnemonics over here. For example, for anaerobes, generally we have can't breathe fresh air. So we have read there are two anaerobes, the C1 and the A1. So A for actinomyces and C for Clostridiae, B and F for bacteroids, and E for Fusobacterium. Then also we have the intra. These are the normal flora. Facultative, spore forming. So there are only two: the bacilli, gram-positive bacilli, and Clostridia. They are only the spore forming bacteria. Then we have the urease producing bacteria overall, and in this one we have the P chunks. In the P chunks, P for Proteus, and then C for Cryptococcus, H for H pylori, urea plasma, then nocardia, Klebsiella, then Streptococcus, staph epidermidis, and saprophyticus. And the next one is intracellular pathogen. So in intracellular pathogen, first discuss the facultative one. There is a mnemonic over here. Some nasty bugs may live facultatively. Okay, so some for salmonella, and nasty for Neisseria. Then B for bugs for Brucella. Then M for Mycobacteria, Listeria, Francisella, and Yersinia pestis. So this facultative one. Then the obligate, which rely on the host ATP are the Rickettsia, Chlamydia, and the last one is Coxiella. So these are pretty much low yield mnemonics, but these mnemonics they help us a lot in detecting the bacteria. These are given in the scenario. So this is all about the classification of gram-positive bacteria and some relevant mnemonics.